So thanks, Savandi, and thanks, Arathi, for organising today, the 2 days. My name's Joel Robertson, and I'm a PhD student at RMIT. I'm going to talk to you today about my ongoing research, which is looking at the interactions between coding and non-coding transcripts in plants using heterogeneous networks. And that's not letting me, yeah, cool. So just a bit of context about the area of network science that we're operating in. We're looking at graphlet counts, and a graphlet is just a small subgraph pattern that might repeat many times throughout a network, and you can count these graphlets and use them to try and characterize your network based on its kind of higher order structure. And there's a number of terms that might be used. Sometimes people call them motifs, sometimes they're called induced subgraphs, but they're kind of interchangeable. And I like to stick to graphlets as the kind of the actual structure or the subgraph, and then like motifs actually just indicates the kind of significant graphlet. But some people might have heard it in a different form. I'm sure it's very well known, but just introducing it. You can technically count any size graphlet in your network, but typically with larger graphs, you're kind of limited to kind of subgraphs of size six or less generally. So graphlet counting in the biological context is pretty common. It's often done on protein to protein interaction networks or gene regulatory networks. And these are kind of relatively small networks with directed edges. And they tend to focus on known interactions that can kind of, or have been experimentally validated, and so you actually know the direction of the interaction. An early example of this graphlet counting on biological networks was kind of one of the first prominent publications that kind of drew attention to the idea. They looked at E. coli transcription regulation networks and kind of identified these feed forward loops as being significant in those networks. And that, I think, excited a lot of people, maybe in the biology community, that they had this kind of mechanism to apply to this ever increasing stream of data that they had, and kind of could condense it down into this more informative output. And that trend towards bigger and bigger data has continued since then as well. There's RNA sequencing these days now can give us pretty comprehensive coverage of everything that is expressed within a cell in terms of the transcriptome. So the question then becomes even more so, how do you deal with that huge amount of data? And one popular approach is to look at your transcription expression data in the form of a co-expression network. So a co-expression network comparatively is very large often, and you may have thousands or tens of thousands of nodes which correspond to the transcripts, and the nodes are connected by edges that represent their co-expression in the data. So it's completely derived from the data, and these edges are undirected. And typically the analysis on a co-expression network involves looking at the community structure, and you form these gene modules that kind of indicate where at the function of an unknown gene or transcript based on the genes that shares a module with. You can look at the graphlet structure here. It's not really done often, probably because smaller graphlets on undirected networks aren't that interesting. There's not many to look at comparatively, but we can kind of induce a more complexity as if it was a directed network if we look at a heterogeneous network where there's different types for the nodes. In this case, they are coding and non-coding transcripts, and I'll just explain what I mean by that for people who don't have much familiarity with molecular biology. So what we typically call a gene is just a section of DNA that codes for a protein. There's actually large parts of DNA that don't do this. Most of the DNA, particularly in humans, doesn't do this, and for a long time that was thought to mean that it was irrelevant or kind of junk and not to be studied. But now we have this increased ability to observe the transcriptome. We understand that these sections are being transcribed and are expressed, and they actually seem to be quite important. And one theory actually holds that they are involved in this kind of language within the cell, where they allow different genes to communicate with each other. And in plants in particular, they've been linked in some early studies to a plant's response to a stress condition. But in general, studying these non-coding transcripts is quite difficult, because even more difficult than in just general genes, because there's no kind of output that you have with a gene, like a protein, where you can see what its kind of purpose is or what it's leading to. So we need to try a different approach, and we use heterogeneous networks to do that, and look at the type graphlets on those networks. So to construct our networks, all we need is a transcription expression profile for each sample, and then a type classification for each of those transcripts. We also want to get rid of transcripts that aren't doing too much across all the samples. So if they're not got a very high expression in general, or they're very stable with their expression, then we want to ignore them. And it's also important then in the inputs to have samples that allow this variation in expression. You want kind of diverse samples, whether that be time-based or condition-based, or even just a high number of replicates. But the most important thing in the step in the construction process is determining how co-expression is decided upon. And in a typical co-expression study where you're looking at the community based kind of analysis, often a variation or some variation of a correlation coefficient is used. It's a bit of a problem here, because that can lead to kind of spurious edges where the correlation is not kind of direct, but based on other nodes that the nodes might be connected to or correlate with. So we want to use a partial correlation measure that isolates that just only the direct interaction between the two nodes in question for an edge. Once we have a constructed network, we then need to count the typed graphlets, and most graphlet counting algorithms can be adapted to look at typed or colored graphlets, just to count type of colored graphlets, but doing that adaptation can add inefficiencies and become a bit prohibitive on larger networks. So we use a modified version of an approach that Robertson laid out that's kind of specifically designed for heterogeneous networks. And it's interesting, it actually uses a per edge approach, iterates over the number of edges, which seems a bit counterintuitive at first, because there's a lot more edges in most networks than there are vertices or nodes. But it makes sense in this context, because when you start with an edge, it gives you the two of the types kind of fixed already, and then you can just look at the neighborhood of the edge in question and just identify the three paths that edge is involved in, and the triangles that is involved in, and then extrapolate from those three node subgraphs or graphlets up to the higher order ones that you want to look at as well. And we in particular are interested in maintaining the order of the types. So for instance, we want to know if in this three path here, we want to know if this is a non-coding or a coding transcript, because it's different structurally to if the non-coding transcript was on the edge of that graphlet. So we use then the orbit of each vertex to maintain type ordering as we are counting the graphlet. And another benefit of this approach is that it gives us a local graphlet count for every edge, and then by extension every single node we can work out the graphlets that just it itself is involved in. And it also allows us to run the whole process in parallel, because each purge process is really independent of the other. So just now looking at some applications of this counting. As I mentioned, the typed graphlets give you a lot more complexity to deal with, even at small lower order graphlet types. So lower order graphlets, so gives us much more of a graphlet profile with which to characterize the network. And a few ways that we can do that are looking at motifs, which is comparing the graphlet profile of our real network to a set of randomized similar networks using a null model, and determine which graphlets are significantly overrepresented or underrepresented. We can also look to compare the two different real networks and look at their graphlet profiles and see how they differ, and that is quite relevant in a biological sense. So say with the plant example that I was mentioning before, you could have two different networks, one based on the stress condition and one based on control condition, and then see how the transcript interactance is changing over that condition. You can use the network motifs that you might discover to try and understand what role the non-coding transcripts are actually playing in these interactions specifically. And similarly, you can look at a specific graphlet type and look at the sub counts based on its type comparisons and try and see how they differ maybe with different type orientations. And lastly, we think that the graphlet counting approach it can actually complement the module detection, help form these communities that then imply functions. So I did want to have some results to show today, but they're kind of preliminary, and there's definitely still a fair few issues there that mean that they kind of would be not really that informative yet. So we're still getting a high skew in our graphlet counts towards highly connected subgraphs, which is associated with the problem I mentioned before with your co-expression measure. And that's kind of to do with the number of samples in the data that we're using at the moment, which is, there's not enough samples for the partial correlation measure to actually rule out the indirect correlations. There's also concerns about how the null model construction might lead to bias in towards specific motifs. And I think that is actually kind of a problem in general with motif detection analysis, but need to kind of look further into that for. And lastly, we just want to make sure that our network and then the graphlets, or the significant graphlets that we might identify, actually have some biological validity. Even though we want the kind of process to be driven purely by the data, kind of to be taken seriously biologically, it needs to match up with some known biological results. So that's all. Thank you to my supervisors, Stephen Davis, Alice Johnson, and Nitin Mantri. And any questions I'll be happy to answer now or via email. And I've just got some references there.